of white matter injury, and it was also important that we were able to have matched controls between all sites, which I'll explain you why in my next slide. Because for this harmonization technique, to implement this harmonization technique, we need a subset of controls that we can match for demographics. Typically, age and sex can be sufficient, and we need at least around 15 to 20 controls. And the way we do this is that we select one of the sites as a reference, and the rest of the sites, we define them as targets for harmonization. And using this subset of controls, we compute some scaling factors for the signal, and the scaling factors are then applied to the remaining data set to harmonize the data. So if we move to the next slide, I'll show you how these differences in diffusion actually look across sites. So if you look here at the top row, what you're seeing are differences between each target site and our reference site. So here the color map, the color in yellow shows you regions of the white matter where there are significant differences between sites. And I stress that these are matched controls between sites. So in theory, if there is no variability due to scanner, we would not observe these differences. But as you see, before harmonization, we have significant differences across the whole brain. But after harmonization, these differences are flattened out. But it's also important to, of course, pay attention to the second side, which is we flatten out these differences, but do we preserve the biological signal of interest? So in the next slide, what I show you here is the average FA values for each patient and controls in the white matter skeleton. So if you can appreciate here, the gray boxes represent the controls and the green ones represent the patients. And you see here results before and after harmonization. And basically what I want to show you here is that before harmonization, you can see that the gray boxes are not aligned, and this is because of the systematic differences across sites. And after harmonization, you can see that controls all fall on the same line. But it's important to notice that also the patients are scaled with the same amount within each site. And this means that effect sizes between groups, between patients and controls within sites are preserved. So we had very minimal change in effect sizes. For the next slide, I'm also showing you here associations with my white matter hyperintensity volume. We observe exactly the same thing. Before and after harmonization, the relationship that we find between diffusion metrics and other markers are maintained. So now we can say we can pull this data into a single larger data set in order to look at the same relation, but with a higher statistical power, which is what I'm showing you in the next slide. If you notice here on the left panel, we have the results before harmonization. And even though this association is still very strong, still very significant, you can see that we have some loss of explained variance. And this is because you can notice that data points from different sites are clearly clustered in separate clouds due to these systematic differences across sites. But after harmonization, the data actually behaves as a single site data. And we're able to explain much more variance in FA with higher statistical power. And here I show you results for white matter hyperintensity volume, the relationship with between this marker and diffusion metrics. But you can also imagine that we could be looking, for example, at relation between diffusion and cognition, which typically have much more subtle effect sizes. So if we move to my next slide, I conclude saying that the method that I show you here, I left it as a black box, but basically this method is very advantageous because we apply it on the raw diffusion signal rather than on individual metrics. So we applied it at the beginning of the processing pipeline. So we don't have to do this multiple times. And this is very advantageous for retrospective studies that want to use already acquired data to answer new research questions. And with this, I conclude my presentation to the next slide, which I would like to thank, of course, everybody that contributed for this study, particularly all the PIs from each group that provided the data. Yeah, and that's it. So thank you. And I'm happy to answer questions and also tell you more about the pros and cons of collaborations. Anybody has questions?
Thank you, Bruno. Thank you so much for your wonderful presentation. There is, I can't see question for now in the chat, but maybe I can start. So you showed that the 5 scanners in your studies, they were from a 3 Tesla MRI.
Mm-hmm.
Do you think that this kind of method can be also applied for to data set that they collected data on a 1.5 Tesla MRI or even lower strength of the scanner?
Yes, in theory it's possible. But of course, we try to harmonize the acquisition first, and when that's not possible, we can apply this method. In theory, it's possible. Everything depends on how you model the diffusion signal, because the diffusion signal is a complex signal. For example, you acquire data of multiple directions, and these directions, they encompass different properties of the signal. In theory, you can harmonize data from different field strengths, but so far we only applied for 3 Tesla. Yeah.
Okay. So we have a question from Joel Ramirez. Bruno, very convincing and impressive result. What is involved with harmonization? For example, is this accomplished computationally or other approach?
Yes, it's computationally. For example, I don't know if you are familiar with statistical harmonization, for example, combat. In combat, what we do is that we use the site as a covariate in the analysis, and we try to remove this. But with this method, we use the properties of the diffusion signal, because the diffusion signal is, you can decompose it in several components which carry different information, and this component is what you try to harmonize across sites. And you do this on the signal and not on the metric per se. So it's basically you're trying to average out, or you're trying to remove the side differences on average in this component in the several components of the signal. I don't know if that replies the question.
So sorry, follow up. I just wanted to ask, so how much data do we need in order to accomplish this?
This method has been published with healthy controls, and the original method, they recommend using at least 15 to 20 controls, 20 healthy controls. And obviously we try to match these controls as much as possible. And when I say matching, I mean mainly age and sex, but you can also think for our types of controls, which are elderly controls, you need to think of other things like white matter hyperintensity volume or brain volumes, and etc. But typically you need around 15 to 20 controls. We tried even with 13, and we had positive results in this case.
Okay. Yes. We have one more question from Dan Tozer. Thank you for your presentation. Does the harmonization rely on having similar B values / number of directions, or is it independent of that?
So here we were still within the regime of the diffusion tensor model. So we had B values of 1000, and other sites also had B values of 1200. And there are ways of scaling the B values, scaling the signal to the same B value. But if you have multi-shell data where you have data with 700, say 1000 and 1500 of B value, you can still harmonize the separate B value separate shells. Yeah, but typically you try to harmonize the acquisition as much as possible, and when that's not possible, you harmonize the signal in that way. Yeah. Okay.
I see that there are other questions in the chat, but I think we should move to the next presenter. So we can take the questions later, or Bruno, if you want, in the meantime, you can reply in the chat.
Yeah, and I'm also happy to reply by email for anybody that wants to.
Yes.
Thank you.
Thank you. So our second speaker is Dr. Julie Autois. Julie Autois will present about the effects of vascular burden on cognition are mediated by atrophy, amyloid, and glucose metabolism, a multi-center mixed cohort of small vessel disease and Alzheimer pathology. Dr. Julie Autois is a postdoctoral researcher at the Sunnybrook Research Institute in Toronto, in Canada, and she will present about a possible mediating mechanism that explain the relation between vascular burden and cognitive impairment. She studied these in a multi-center mixed cohort of patients with the small vessel disease and Alzheimer's pathology as a part of the MITNEC 6 study. This study was conducted in several centers throughout Canada, so a big national collaboration. Julie will also tell us about her experience in working with the data from these different centers. Julie, the floor is yours.
Thank you very much for this introduction, and thank you for inviting me to speak today. I can go to the next slide, thank you. I have no disclosures, next. So small vessel disease often coexists with the pathologic and clinical diagnosis of AD. For example, in this study, in 447 individuals, AD mixed with vascular pathology was most commonly observed in over 75% of the probable AD cases. Next. The most widely used biomarker of small vessel disease is white matter hyperintensities detected on MRI. And emerging studies have shown a relationship between white matter hyperintensities and cognitive decline, amyloid accumulation, and cortical atrophy. However, knowledge is still limited regarding the effects in patients of more extreme endophenotypes with evidence of significant vascular burden in addition to AD pathology. Next. As such, the Canadian MITNIC C6 or the C6 project of the Medical Imaging Trials Network of Canada, which has been recognized as part of the worldwide ADNI, was developed, which included real-world patients from dementia and stroke prevention clinics without overt stroke that presented with moderate to severe white matter disease, and they were recruited across 9 sites in Canada. Next. The main objective of the MITNIC C6 was to quantify cross-sectional and longitudinal alterations in brain amyloid, glucose metabolism, structure, and cognition, and subjects were followed for 24 months. Next. So various data was acquired, including neuropsychology, imaging markers such as amyloid and FDG PET, and 3 Tesla structural MRI, and we also did blood sampling. So this MITNIC C6 project exemplifies great collaborative efforts across Canadian research institutes that focus on dementia and SVD. Next, please. So in light of this collaborative session, I will just zoom in a bit more on our collaborative aspects. So first, our collab was established because of challenges that were associated with our strict inclusion criteria, which was moderate to severe white matter disease. Second, our collab came about through existing relationships of Dr. Sandra Black from Sunnybrook with other research sites that study stroke and dementia across Canada. Third, the challenges of our collaboration were mainly related to the PET tracer, specifically the local manufacturing regulations and approvals. And also we had challenging inclusion criteria where we initially only included severe white matter disease, but this had to be relaxed over time to also include moderate. And last, in an effort to harmonize our data acquisition across sites, we applied common PET and MRI protocols, and we performed the image QC, archiving, and processing at one central site in Sunnybrook. Next. So based on this MITNIC C6 data, the primary objective of our study was to investigate the association between white matter hyperintensities and cognitive impairment. And secondly, we investigated the mediating roles of amyloid and neurodegeneration within this relationship between hyperintensities and cognition. Next, please. So for the methods, we used amyloid PET, FDG PET, and cortical thickness in AD signature regions as displayed here on the images with the corresponding references to the papers. And for the cognitive impairment outcome, we assessed semantic fluency based on animal naming, processing speed based on the TRAILS-A test, and executive function based on the TRAILS-B test. Next, please. Our pipelines were adapted towards multi-site data, including for the PET, the smoothing to a common PET resolution and the use of a common reference region for SUVR quantification. And for MRI, we used an enhanced resurfer workflow for patients with SPD that was developed at our lab, and also fully automated segmentation pipelines to quantify white matter hyperintensity volumes. So harmonization in general is an important aspect of dealing with multi-site data. And for me personally, being an imaging researcher, it was challenging, but also very enriching to learn how to process this multi-site data. Next, please. So this slide shows our demographics, where we included 60 subjects from the MITNIC C6 who had moderate to extensive white matter hyperintensity volumes. And we also matched 60 subjects from ADNI who were normal control or early MCI with mild to moderate hyperintensity volumes. Next. In the MITNIC cohort, the global amyloid was significantly greater and thickness lower compared to the ADNI. Next, please. In addition, the MITNIC C6 subjects performed worse on the cognitive domains, including semantic fluency, executive function, and processing speed. Next, please. So as a result, including all the subjects of both the ADNI and the MITNIC, we found that increased white matter hyperintensity volumes were significantly associated with cognitive impairments on the different domains that we tested. Next. So this relationship was mostly due to the strong association between hyperintensity volumes and cortical atrophy, here nicely shown for semantic fluency as our cognitive outcome. In addition, we also found a significant indirect effect of hyperintensities on cognition via amyloid PET, but this PET had a lower effect size. Next, please. Then the path through amyloid followed by hypometabolism had the lowest effect size, though was still significant. Importantly, when the AD signature regions where we evaluated our 3 imaging mediators were substituted by a more focal temporal meta-ROI, then the significant mediation effects increased from 68% to 74% of the total effect, which also highlights the vulnerability of specifically the temporal lobe to vascular associated atrophy. Next, please. When the semantic fluency was then substituted by executive function as our cognitive outcome, similar paths through atrophy and amyloid were found, though generally they had a lower effect size, and the path through FDG was not significant. Also here, the mediating effects were stronger when a temporal meta-ROI was applied. Next, please. So in conclusion, in a cohort that represents the more common AD population with vascular comorbidities, next, we found that hyperintensities were associated with poor cognition. Next. This relationship was mainly due to the amyloid-independent effects of hyperintensities on cortical atrophy, subsequently causing the cognitive loss, and to a lesser extent through the effects of hyperintensities on amyloid or metabolism. Next, please. Our study highlights the importance of developing therapeutic approaches that target vascular factors in addition to AD pathology, but which are also safe to administer in a more common AD population. Next, please. So finally, I would like to thank all the patients and caregivers, my supervisors from Sunnybrook, Dr. Black and Dr. Gubran, and all collaborators across Canada. And we also like to thank our funding sources and Eli Lilly, who kindly provided the Floor Beta Pierre tracer. Thank you.
Thank you, Julie, for this very interesting work. So please ask questions in the chat. I have a question actually, because your model, they show that basically the path via amyloid doesn't have a strong effect, but through the most of the effect is in the path through cortical thickness. Mm-hmm. So more like overall neurodegeneration. So my question is, how do you interpret this? Is because it could be that maybe it's not so much Alzheimer related neurodegeneration that is playing a role, but maybe other kind of neurodegeneration, or is it something else?
Yeah, that is a good question. So we have a mixed cohort of AD and vascular pathology. So there may be definitely other factors at play that influence this relationship for sure. I think the fact that we also see that the temporal lobe actually has a very important factor may also highlight, in AD, for example, the temporal lobe plays like a crucial role, is very vulnerable to amyloid and tau pathology. So this may have worsened the effects or the path through atrophy. And also there could be secondary widespread neurodegenerative effects on the gray matter in the temporal lobe by damaging, by the fact that the white matter hyperintensities may damage specific white matter tracks that serve these temporal cortical regions.
Okay, thank you. Are you planning to follow up, because I saw that the study design now was cross-sectional, are you planning to follow up and this cohort and see what happens longitudinally?
Mm-hmm. Yeah, for sure. We are acquiring longitudinal data over 24 months. And one of the goals will definitely be to look at whether hyperintensity burden at baseline predicts amyloid accumulation over time. Because this has definitely also been shown in the literature. There are a couple of ADNI papers that have shown this, but as you know, with the ADNI cohort, the subjects that were included were mainly stratified for AD pathology, but then they don't have a large amount of SPB. They were excluded if they had a lot of SPB. So therefore, I think our cohort will definitely contribute to better insights in this relationship between hyperintensities and AD pathology in a cohort that also includes more SVD burden, which is actually more representative of the common AD population.
Wonderful. Thank you so much, Julie. And now we can move on to the next two speakers that will be introduced by Dr. Hilde van den Brick. So Hilde, go ahead.
Thank you, Anna. I'm not yet a doctor, but thank you.
It's okay.
It's my pleasure to introduce the next speaker. It's Ben Lam. Ben Lam is a postdoctoral research fellow at the Centre for Healthy Brain Aging at the University of New South Wales in Sydney in Australia. So thank you for still being here with us despite the time difference, Ben. Ben will present risk factors for onset of post-stroke depression, which are results from the StrokeCo Consortium. This project is a great example of how collaboration in the StrokeCo Consortium allows to assess research questions in diverse ethno-regional groups. So we're excited to hear more about it. Ben, please go ahead.
Thank you for your introduction. So good evening. It's actually 10:30 in Sydney. So it's a virtual background. So I pretend that I'm in Europe with you guys. So thanks for the opportunity to present this international collaborative research looking at risk factors for post-stroke depression in diverse ethno-regional groups. Next, please. So we know about depression is very common among stroke patients. About 1/3 of the patients experience depression and other mood disorders after their stroke onset. And there are various adverse effects of PSD on stroke recovery. Next. And so the current research project tried to use individual patient data from an international consortium to investigate risk factors that predict incident PSD, and hopefully can inform screening and treatment of PSD in these stroke survivors. Next. Next. You can, yeah, thank you. So there have been a number of risk factors that were identified and examined in previous research, including demographics like young age, female, lower education, which show mixed finding in the literature. And then there are also stroke characteristics. One of the first strong predictor of PSD is severity of stroke. And other factors like type of stroke, site of stroke have also been examined, but with mixed findings in the literature. Vascular risk factors and diseases have also been examined, but with no systematic associations with PSD. Next, please. And 2 more clicks, yes. And the other strong predictors of PSD include history of depression, of course, as well as past history of stroke stroke, impaired functioning, as well as impaired cognitive cognition. Next, thanks. So in this study, we used the data from the Stroke and Cognition Consortium, StrokeCOC, which is an international consortium for neurocognitive disorders in relation to cerebrovascular disease. To understand these risk factors for PSD, we analyzed the individual patient data so that we can apply standardized data and statistical methods like classification of depressed individuals, as well as to control for the same set of covariates across studies. For these international data, we are also able to examine these effects in diverse ethno-regional groups, which allow us to test for generalizability as well as to explain the heterogeneity across these different places. And StrokeCOC is an expanding consortium with currently about 34 participating studies covering about 5 continents. Next, please. Thank you. So currently, for this current project, there are 9 contributing studies, which are from European countries, Asian countries, the States, as well as Australia. And the total sample size for this project from this contributing study is about 1,700 stroke patients. Next, please. So we follow previous protocol to harmonize the data, including using given clinical cutoffs for the different depression measures used in this study to classify depressed versus no depression. Next, thank you. And we also classify assessments on functional impairment, cognitive impairment, as well as stroke severity. Next. Other factors that we assessed include stroke subtypes, site of stroke, different vascular risk and disease factors, history of depression, past history of stroke, as well as age, sex, and education. Next, please. So we conduct mixed effects cost regression to look at these risk factors in the prediction of incident PSD. And in all the models, we control for age, sex, and education. And we first examine univariable models for each individual risk factor. Then for those that were significant, then we include all of them in a multivariate model. And then we use multiple imputation for imputing all the missing data in the studies. Next. So in this sample of stroke patients, about 35% develop PSD during the average follow-up of 2 years. And then the median time for the first diagnosed depression is about 5 months, which is consistent with the literature. The incident rate of PSD is estimated to be about 200 cases per 1,000 person years. So about 1 in 5 stroke patients will develop PSD in a year. Next, thanks. So in this table on the under the risk factor column, these are the risk factors that were significant as univariable risk factors. Except age, sex, and education, which were there in all the models. And the subtypes of stroke, site of stroke, as well as the different vascular factors were not significant in the univariable model, so they were not here. So after putting all these significant risk factors into a multivariable model, we can see that females, lower education, history of depression, functional impairment, and cognitive impairments were all significant risk factors for PSD after controlling for each other. Next, please. We also examined if these effects differ across these different study regions. However, we didn't find any significant main effect of region or any interaction effect with the risk factors. Next, please. So in summary, we find these are the significant and strong predictors of PSD across these different studies, including female, lower education, depression history, poor functioning, and poor cognition. of these risk factors, consistent with the literature, but then confirmed in these analysis that they have unique effects on predicting incident PSD. And further development of these studies include examining short-term versus long-term effects of these risk factors, as well as exploring the interactions among these different risk factors. Next, please. So finally, I would like to talk a little bit about the advantages and challenges for using stroke to understand cardiovascular diseases, and if you have any research questions that you think may benefit from this use of data, please feel free to approach us. So some of the advantages include having relatively large sample size and high statistical power. And we can also examine regional differences and similarities because these are international data. But then challenges include the availability of data. So not all the measures may be available in every study. And also we need to examine the heterogeneity across these different studies. Next, please. So thank you. If you have any questions about this present, feel free to approach me through this email. And if you're interested in the use of these stroke data and to discuss your own research ideas, you are also welcome to contact the principal investigator and coordinator for this construction. Thank you.
Thank you, Ben, also for that great invitation that you just finished off with. Very nice to see what you can do with such a big data set. And I already see a question coming in the chat. Yep. First, your compliments from Joel. Whoa, Ben, very massive undertaking, for sure. I'm quite shocked that laterality was not significant. Were the patients generally more right or left, considering how difficult it is to perform cognitive testing, etc. on the left?
So combining all the data, the proportion, it's about the same, but then there are a small proportion of people who are affected on both sides. So the findings on the sign of stroke on these post-stroke mood disorders are very mixed in the literature. There are some earlier findings showing there are differences in terms of the different location and the size, but then some recent replication studies do not support these results. So it's important to conduct these more large-scale study to look at the effects of these different risk factors.
Thank you. And very briefly, maybe if you can answer briefly a question for myself. You showed that there are no you showed that there are no differences in the risk of post-stroke cognitive depression over different regions, but you also showed that education is an important independent predictor of post-stroke depression. So I was wondering, because we also saw that education seemed different in different regions, how do I interpret these two findings together? So
it's a very good question. This speaks to one of the limitations of this research. So we do see at the individual level, people with lower educations, they are more likely to develop post-stroke depression. But then across these different regions, we do not see any differences. But you can see that most of these studies are from high and and about middle income countries. I would say most of them are from high income countries. And so it limits the comparison to this group of countries. And it's important for future research to include more low and middle income countries in this study. And like that, because of this restriction of range, we are not able to see any differences across these regions. But maybe with more studies from these other regions, then we may be able to further examine this effect.
I see. So more data needed for stroke. Thank you, Ben. I'd like to continue to the next and final speaker of this session. So it's my pleasure now to introduce to you Dr. Guido Gravenstein. He's a postdoctoral researcher in the CADASIL research group of the Leiden University Medical Center in the Netherlands. Guido specializes in CADASIL and will show you that specific Notch3 mutations are associated with different Notch3 aggregation loads in CADASIL patients. With degrees in both medicine and biomedical sciences, Guido has a strong affiliation for translational research, and his presentation is a great example of how we can benefit from collaborations between different disciplines. The floor is yours, Guido.
Yeah, thanks a lot for this introduction, and thanks for allowing me to present here. So as Hilde said, I have a particular interest in translational research. So I was quite lucky and happy that I could join the CADASIL research group on the next slide. When I started my PhD, and the nice thing about the Leiden CADASIL research group is that we really focus all the way from molecular biology to neuropsychology. We're really going from bench to bedside, and we can because we are focused around CADASIL. And on the next slide, just a quick recap that CADASIL is a hereditary small vessel disease where typically patients develop mid-adult age stroke, which often become recurrent, and it ultimately leads to vascular dementia. CADASIL is always caused by a mutation in the NOTCH3 gene on the extracellular domain of the NOTCH3 gene, and this results in NOTCH3 aggregation. And because NOTCH3 is mainly expressed in the vascular smooth muscle cells in the vessel walls, this leads to an arteriopathy, either directly or indirectly. And this leads to dysregulation of blood flow to the brain, white matter hyperintensities, and ultimately also lectures. And although patients only exhibit symptoms of the brain, so to say, the NOTCH3 aggregation is systemic, which allows us to study NOTCH3 aggregation in skin biopsies, because then we can take a look at skin blood vessels. And we can do this in two ways. On the first way is using immunohistochemistry on the left side in the figure. That we can stain NOTCH3 using an antibody, and we see the typical NOTCH3 granules, which are an entity of NOTCH3 aggregation. But we can also take a look at electron microscopy, zooming in a bit further. And there we see surrounding the neural cells or surrounding the vascular smooth muscle cells in red, we see these gray blobs, which are called granular osmophilic material, or GOM in short. And this is another entity of NOTCH3 aggregation. And because NOTCH3 aggregation is such a central, plays such a central role in disease pathophysiology, in the beginning of my PhD project, we wanted to assess whether we could link pathology to disease severity. We wanted to see whether there's an association between NOTCH3 aggregation and disease severity. And we tried it in my first year. Next, please. And we failed. And that was a bit disappointing. No matter how hard we tried, we didn't see any nice correlation. We didn't get any significant results. So we abandoned the project for a while. But then over the years in our research group, a finding was done regarding the genetics part of the CADASIL research. And that allowed us to change perspective a bit on the initial research question. And that's what I'd like to tell you today. So regarding this genetics finding, in our group, the first phenotype correlation was found in CADASIL, and it basically says that NOTCH3 mutations located in the first EGFR domains of the NOTCH3 gene, they predispose to a much more severe phenotype of CADASIL, basically the classical phenotype of CADASIL, rather than if a mutation is located in EGFR domain 7 to 34. Then on average, it predisposes to a much milder phenotype. So we thought at that point, well, now we know this, does this actually underline NOTCH3 aggregation? So in the first year of my postdoc, our research question became, next please, is NOTCH3 aggregation associated with NOTCH3 variant position? So that's what we wanted to find out, and we took a look at the DVNAS cohort, which is a CADASIL cohort recruited in our research center. And we took a sample of about 25 patients with either a 1 to 6 mutation or a 7 to 34 mutation. And we included both patients with a severe, but also with a milder phenotype. And all patients were age and sex matched in order to correct for that. All patients got a full workup, including a skin biopsy. And this allowed us to check for NOTCH3 aggregation in skin, which I depicted on the next slide. And well, we can do that in two ways. We can assess NOTCH3 aggregation in skin blood vessels using immunohistochemistry. Therefore, we use the NOTCH3 score, where we basically calculate the area which is positive for NOTCH3 aggregation over the total vessel wall area. But we also take a look at the electron microscopy analysis on the next slide. Yeah, and well, this is manual work. So in the first year of my PhD first year of my postdoc project, we assessed over 500 blood vessels and counted 400 gums. And we really hope to find finally something that was nice and perhaps even statistically significant. And on the next slide, you can already see that we found it, because we immediately identified that patients with a 1 to 6 mutation actually have much higher NUTS3 aggregation levels than 7 to 34 patients. And although I only show you 2 examples here, we assessed many vessels per individual. And in the next graph, each dot represents the average of one individual. And we see a massive difference between 1 to 6 mutations and 7 to 34 mutations. So we were very happy to find it, but we wanted to check it further. So we also, well, we took a look at the electron microscopy data on the next slide. And there, basically, we found the same results. We found very high gum counts in the 1 to 6 patients compared to the 7 to 34 patients. Although still there is quite a large variability, even with a couple of patients having no gum at all in the 7 to 34. Well, one major question now, of course, is, well, this is going on in the skin. What is going on in the brain? Because that is the place where the actual disease takes place. Well, unfortunately, we cannot take brain biopsies, but we had 6 brains of diseased CADASIL patients available. And on the next slide, well, I show you that we found the same trend in these 6 individuals, but yeah, obviously we cannot do any statistics on this. But well, we have the feeling that this is actually a true thing. So in summary, we show that NUTS3 aggregation is indeed higher in EGFR 1 to 6 patients than in patients with a mutation in EGFR 7 to 34. And currently, we can only speculate as to why this is. But one logical explanation might be that these mutations are simply more exposed to the extracellular matrix and are more likely to recruit extracellular proteins such as HRA1, as Professor Dick this morning said. But still, we see a large variability within the groups. It might be that we need to further delineate the effect of the variant position, but it might also be that more modifiers are present for NUTS3 aggregation we currently don't know. Well, then coming back to the initial question, what is the relation with actual disease severity? Well, that's something we'd like to study in the full DFINAS cohort of almost 200 patients. But we already took a quick look in our small data set, which I showed you today. If you have another 7 minutes left, you can look it up in our paper because it recently got published. Next, please. You see the title. But I think the most important conclusion for this talk is actually that really a finding in one aspect of the field can really contribute to a better research question or new ideas in your own field. And with that, I'd like to thank the research group and all our collaborators, and of course you for listening. Thank you so much, Guido. Oh, I think we can. This is just in case there is a question. Thank you. So not like a plenty number of patients to study, but a huge number of blood vessels to go in and count. What an effort. Very nice and very convincing work. If there's any questions, please put them in the chat. Indeed, you already alluded to it at the end, but I wanted to ask, because I also read it a bit in your abstract, can you already say a little bit something about the relationship with disease severity, or is that too difficult to say in a minute? I think we can. Perhaps we can go to the slide that was actually just shown, because that's the data we have. So we basically had 2 subgroups. We had either a patient with a severe phenotype defined as having 2 legumes or more, or with a milder phenotype. It's in number 61, please. So, and then if we split it in these 2 groups, please go to slide 61 if possible. Yeah, that's great. So here we see the same data, but then split it between severely and mildly affected patients. And what is immediately visible is that there seems to be a kind of a trend with disease severity in the EGFR 7 to 34 group, that it seems to be more NUTS3 aggregation there. in the severe patients compared to the mild patients. But then when we take a look at the effect in the 1 to 6 group, well, I think we need a larger sample size there. But still, what is I think what is visible here is that the effect of mutation position is much larger than the effect of disease severity. So that's something we are trying to find out currently in our lab.
Very cool. Also, thank you for so nicely showing us the asset and the benefits of doing the translational work. I see a question also coming in the chat. Hi, Guido, do you know if Notch3 aggregates are found in specific cell types?
Yeah, we do know. So Notch3 is mainly expressed in vascular muscle cells, in the neural cells, I should say, surrounding the endothelial cells. So these Notch3 aggregates are located extracellularly around these vascular muscle cells and pericytes. We found that, well, when we take a look at electron microscopy, that we found a lot of GOMs surrounding the vascular muscle cells. We found lesser GOMs surrounding the pericytes in the capillaries, and even lesser lower GOM count in the venules, but we did see some GOM in the venules surrounding the neural cells. I hope that answers the question.
I think it does. I think it does. Thank you so much. Yeah, well, there you got your confirmation. Yeah. If there are no further questions, I'll wait a little bit. Oh yeah, there's still one coming in. Do we know why certain brain regions are more susceptible?
Yeah,
I think as you're referring to the white matter hyperintensity lesions that we see in specific patterns in CADASIL, I think.
Yeah, I find that a bit hard to answer. I don't have a clear-cut answer right away. But what we do know regarding the pathology is that especially the penetrating arteries are mostly affected. Well, there is something there in the vasculature anatomy that makes these vessels susceptible over vessels in other places in the body. But yeah, the clear-cut answer, I don't know.
The key question maybe to answer.
I think yes.
I don't think there are any further questions. And with that, I'd like to take some time and take a few words to wrap up the session. So first of all, thank you very much to all the 4 speakers for your great presentations. We have seen 4 presentations with important findings that could only be acquired through collaborations, the theme of our session. We have seen examples of collaboration over different disciplines, but also collaboration between different sites to acquire data, collaboration to pool already existing data sets, to allow to answer questions that require larger sample sizes. So we have seen that each type of collaboration also has its own challenges, and we should learn from each other's experiences, I think. Of course, collaborations often spark when we meet and when we talk to each other, which is difficult to do in current times. However, a good start is also to go out there and present your work and see what others are doing, which we are doing these days. That's also why we are very happy and thankful that the VASCO organizing committee also allocated so much time for presentations by early career investigators so that we can also connect and also see what everybody is doing. And also thank you that Anna and I got the opportunity to organize this specific session. Then lastly, Anna and I have a few plans in the making with which we hope to further help you in finding each other as early career investigators. But also, should you have any ideas or think that we can help you in any way, please reach out to us. You can find our contact details on the VASCO website. There's an early career investigator page. Again, please also connect with us on LinkedIn. I think that's it. Anna, do you have any last words?
No, besides, as you already said, thank you so much for this opportunity for the 2nd year in a row. So thank you so much. And I have added now in the chat our LinkedIn and Twitter account. So please follow them, because that is where we will also announce updates on the next activities that are in the pipelines. So yes, thank you so much to all the presenters. You've done a very great job, guys, in presenting your work and also following our requests on the team. So thank you so much.
Thank
and
you.
thank
Back
you,
to you,
Hilde,
Raj.
to you.